Wonderful. Good afternoon. Thank you to ILSI for inviting me to present this afternoon, and thank you to Drs. Connie Weaver and Dr. Joanna Dwyer for nominating me to provide this talk to you today. The paper that I'm presenting to you today was a major collaborative effort between the University of Massachusetts Lowell, the U.S. Department of Agriculture, Human Nutrition Research Center on Aging at Tufts University, and the University of Oslo in Norway. And I'd like to acknowledge my colleagues that worked so hard on this talk. My lab is interested in identifying nutritional interventions that would be effective in forestalling age-related losses of muscle and bone health. This paper today focuses on osteoporosis or low bone mass, and to me, it's astonishing at the consequences that can occur from osteoporosis or an osteoporotic-related fracture. Of those able to walk before fracture, only 50% can walk again following a hip fracture, and there's 10 to 20% excess mortality rate following a hip fracture. The prevalence of this disease is great worldwide, where 1 in 3 women and 1 in 5 men over the age of 50 will experience one of those osteoporotic-related fractures. And in Europe, the disability due to osteoporosis is greater than that caused by cancers. Interestingly, the prevalence of osteoporosis is different across individuals. For example, you'll find that women have a much higher prevalence of osteoporosis compared to men. And recent research from our group has indicated that Hispanic adults, particularly, contrary to previous belief, have equal to, or particularly in men, greater prevalence of osteoporosis compared to non-Hispanic white and Black counterparts. So therefore, it's important to identify mechanisms of which this disease progresses and nutritional interventions that are specifically culturally tailored and sex-specific. Fruit and vegetable intake, a dietary component across the world, is a potential modifiable factor to prevent osteoporosis. And human subjects research presented on this slide has shown that fruit and vegetable intake is associated with reduced loss of bone over time and reduced risk of osteoporotic-related fractures. Potential mechanisms include a direct effect through phytochemicals, fiber, and micronutrients that are provided by fruit and vegetables on bone health, and also indirect effects through modulating the gut microbiota and certain metabolomic pathways such as reduced inflammation and oxidative stress, all which is positive to bone health. So therefore, the primary objective of this paper was to examine untargeted associations between fruit and vegetable intakes and metabolomic profiles in a cross-sectional cohort of Puerto Rican adults with and without osteoporosis. We used data from the Boston Puerto Rican Osteoporosis Study. 600 individuals presented data on diet, sociodemographic, and health measures. Bone health was assessed by DEXA scan, and metabolomics was measured from plasma blood. 525 plasma metabolites passed quality control in this study. Average age of participants in this cohort was 60 years with a range of 46 to 79. They were generally physically inactive, and 50% had an 8th grade education or less. 8 to 10% presented with the disease, osteoporosis, and on the whole, they generally presented with low fruit and vegetable intake of about 2 to 3 servings per day. We did demonstrate that fruit and vegetable intake, both as servings per day or variety of the types of fruits and vegetables consumed across the day, were both significantly associated with reduced prevalence of osteoporosis in both men and women. Next, we identified metabolites that were significantly related to fruit and vegetable intake following multiple comparisons corrections. And you'll see that we identified in green here 104 metabolites that were significantly related to fruit and vegetable intake, although only 7 of them were shared across men and women. The majority of the metabolites, 12% of them in women, were related to fruit and vegetable intake, and 7.2% of metabolites were related to fruit and vegetable intake in men, and those metabolites were on the majority different. So then we asked, okay, although most of these metabolites are different across men and women, are perhaps the classes of these metabolites overlapping? Are there any similarities? And we did find a couple groupings or classes of similarities. I'm giving one example on the screen here. Carnitine and fatty acid related metabolites, you'll see to the right of the positive beta coefficient, were positively related to fruit and vegetable intake, although each of these shapes here indicate that they were separate metabolites or different metabolites for men and women, but they did belong to a similar class or showing potentially a similar pattern of relationship. We also identified metabolites that were significantly related to the disease state or having osteoporosis, but again, these metabolites were vastly different between men and women. About a third of the metabolites in women were part of the steroid hormone metabolism pathway, which were associated with lower prevalence of osteoporosis. And in men, about 25% of the identified metabolites were branched chain amino acid metabolites, which were associated with higher prevalence to osteoporosis, which was surprising to us. There are only 2 overlapping metabolites across the sexes. The first one is a byproduct of high temperature cooking, for example, frying vegetables, which is not uncommon in a Puerto Rican cohort. And the second one being a precursor for testosterone. And again, I'd really like to highlight that not only were the metabolites that we identified different between men and women, but oftentimes the direction of association was also different. So here, this precursor for testosterone, in women, its presence was related with lower odds of having osteoporosis, yet in men, this precursor was associated with higher prevalence of the disease. Lastly, we took all of these individual fruits and vegetables that we assessed by a food frequency questionnaire in this population, and we used principal components analysis to group them by nutrient similarity or perhaps pattern of intake culturally. And we identified 6 food groups that are identified here in the center of the screen. And although some of these food groups were related to metabolites specifically in women, we did observe some patterns. So for example, berries and melons were related to reduced prevalence of the disease in both men and women, yet potentially through different metabolites for men and women. And the same with dark leafy green vegetables, which appeared to be protective against the prevalence of the disease, yet potentially through different metabolites. So through this work, we were able to identify metabolites that were related to fruit and vegetable intake. We showed that high fruit and vegetable intakes were inversely related to osteoporosis in this Hispanic population. We observed that fruit and vegetable derived metabolites and pathways in relation to osteoporosis were sex specific. And lastly, that green leafy vegetables and berries and melon was protective of bone via osteoporosis specific, yet different metabolites in men and women. Future work in this area, I think there's great opportunity for us to continue to identify other food groups or potentially culturally tailored dietary patterns that are associated with disease outcomes, and how are they individualized? How are these pathways individualized across the sexes, different ethnic cohorts, for example? There's a need for randomized controlled trials to provide these food groups to individuals to then test change in metabolism over time, and again, to continue to observe potential sex differences. Yet I think there are also numerous challenges to overcome. So for example, bringing all of the omics together. How does metabolomics fit with changes that will also likely be observed in the gut microbiome and within the context of genetic variation across different sex and ethnic groups? So therefore, I believe the approach must be multidisciplinary, which is why I'm thrilled to be here with you today and looking forward to the discussion this afternoon. Thank you.